Well, we work in a fantastic field and we're at a really exciting time in eye research because building on some of the knowledge that we've gained about how the eye works over the years, we're now starting to turn that into treatments for conditions that were never previously treatable.
Technology is transforming eye research. We're seeing advances in imaging technologies which mean that we can see aspects of the eye that we've never been able to see before.
My research involves using artificial intelligence to early detect the signs of a condition known as keratoconus. I hope that my research would be able to improve the quality of life of these patients.
The research that I do is based around trying to predict who's at highest risk of progressing from the early stages of macular degeneration to the later stages, which can have a real serious effect on someone's vision and ability to see.
For dry macular degeneration, where cells are dying, we currently have no treatment. And what we've been particularly interested in is trying to slow the progression from the earlier stages to late vision-threatening disease. And at the moment we have no treatment. But I think we're on the cusp very much of having treatment.
Finding new treatments is what we are here to do, and there have been real advances in developing new treatments for vision loss and blindness, but there are still patients that we can't treat, and that's the focus of our work. We want to slow down vision loss in those patients, and we want to restore the vision that they've lost to improve the quality of life.
My research is in glaucoma, and recently we've been investigating vitamin B3 to look at whether you can protect nerve cells and prevent their damage to prevent vision loss in glaucoma. Current treatments for glaucoma are all targeted at lowering eye pressure. However, we know that some people can still progress and lose vision despite treatment. So new therapies to help these people are always important.
Clinical trials are important because they are the most scientific way of making sure that a treatment actually works and is safe.
Well, gene therapy can be used in a whole variety of different ways in the eye. And in my work, developing new treatments for glaucoma, we use gene therapy to make the optic nerve more resistant to injury, and ideally that will preserve vision of patients and hopefully also restore vision when patients have lost it.
My research is trying to produce a tissue-engineered cornea.
We're going to use stem cells, which are created from either skin or blood cells, and use those to build corneal transplants in the laboratory. About 12 million people worldwide are waiting on a donor cornea and can't get one.
My hope for the research is that we can provide corneal donation tissue-engineered cornea to everyone in the world who needs it.
We have kept our research going, our clinical research, our lab-based research, the whole way through the pandemic, and now that we're coming out the other side of that, we're starting to expand and to grow. So we've got some pretty exciting opportunities to build on the success we have as the largest clinical trial centre for vision and eye research in Australia, and make ourselves even more capable into the future so that we can help bring new therapies to patients.
When you do research, you not only help every single person that you see in the room, you actually have the potential of improving the life or the vision of people around the world. That possibility of really making a difference to so many people is what I think drives most of us here at CERA.
I'm one of the deputy directors here at CERA. I'm an ophthalmologist. I'm an associate professor at the University of Melbourne, and I lead the ophthalmic neuroscience team at CERA. It's my honour tonight to pay respects to the traditional custodians on the lands on which we're meeting today. I note that these are the lands of the Wurundjeri people. I pay my respects to Woi Wurrung elders past, present and emerging, and to elders of the Kulin Nation. I'd also like to acknowledge that Aboriginal and Torres Strait Islander peoples are disproportionately affected by vision loss and blindness. CERA and our partners at the Royal Victorian Eye and Ear Hospital and the University of Melbourne are committed to closing the gap in eye health. Before we begin, I'd like to address just a couple of housekeeping matters. Toilets are located behind the auditorium. To find the toilets, just exit via the door to the left, turn to the right and walk through the foyer, and you'll see a sign to the toilets just before the blue doors. In the unlikely event that we need to leave the premises in the case of an emergency, you can see the exit signs there on both sides and then up the stairs. Should evacuation be required, please make your way to those exits, and there are stairwells in the lift foyers. So for members of the audience who can't see me, I'm standing at a lectern to the left side of the auditorium at the front of the room. There's a screen to my left. It's displaying an image of me and my title. I'm wearing a navy suit, a white shirt and a tie, and I've just realised that description makes me sound somewhat like a naval officer. It's a great honour to welcome you here tonight for the 14th Annual Gerard Crock Lecture. And it's so great to see so many friends and colleagues in the audience, and I think it's a testament to the community that we have here at CERA, the sort of level of hubbub and conversation as we're all sitting here waiting for tonight to start, and I think that's something we really treasure at CERA. So I'd like to welcome you all, and in particular I'd like to welcome Professor Duncan Maskell, Vice-Chancellor of the University of Melbourne, research colleagues from the Faculty of Medicine, Dentistry and Health Sciences at the University. I'd also like to welcome Hospital Board Director Dr Susan Srinas, Brendan Gardner, CEO of the Royal Victorian Eye and Ear Hospital, and members of his executive team. I'd also like to welcome CERA Board Directors. We have here tonight our Chair, Olivia Hilton, Nuala Kilgillan, Sawani Dharmalingam, Simon Bruin, who's also on the Hospital Board, and we also have Professor Andrew Cuthbertson, AO, who's Deputy Chancellor of the University as well. I'd like to also extend a warm welcome to Mr Alan Myers, AC KC, who's former Chancellor of the University of Melbourne and one letter shy of a fantastic rock band. I'd also like to welcome Professor Hugh Taylor AC, founding Managing Director of CERA and real champion of Indigenous eye health and two-time presenter of the Crock Lecture. In the audience this evening, I'm really pleased to see that we have colleagues from Breakthrough Victoria who've provided critical support to CERA's new clinical trial centre that we'll hear about shortly. I'd also like to extend a warm welcome to our valued philanthropic supporters, research participants and our dedicated staff. Importantly, it's a privilege to welcome members of the Crock family who are really here tonight to honour Professor Gerard Crock's legacy as a champion of eye research and as an exemplary clinician. We, of course, extend our heartfelt condolences to the family on the loss of Jackie, Gerard's beloved wife, who passed away last year. On screen, there's an image of Jackie and the family at the 2019 Crock Lecture, and we'll certainly miss having Jackie at the lecture tonight. So it's particularly fitting that tonight's Gerard Crock Lecture is being held here at the Royal Victorian Eye and Ear Hospital, and also that it's being presented by the current Ringland Anderson Professor of Ophthalmology, my colleague and friend, CERA Managing Director, Professor Keith Martin. Keith follows in the footsteps of Gerard Crock, who in 1963 was appointed the first Ringland Anderson Professor of Ophthalmology at the age of 34. His appointment was the first medical specialty chair of any discipline in Australia, and he was only the second chair of ophthalmology in the British Commonwealth. To have achieved this level of recognition by that young age, it just sort of gives you a sense of the measure of the man that he was. He established the university's Department of Ophthalmology and much of the Royal Victorian Eye and Ear Hospital as you see it today. His commitment to discovering new and improved ways to treat and prevent eye disease have left a very tangible and enduring legacy. Gerard's niece, Associate Professor Carmel Crock, OAM, is Director of the Emergency Department here at the Eye and Ear Hospital, and she's a great friend and supporter of CERA. So as the beneficiaries and custodians of this legacy, we at CERA, the University of Melbourne and the Royal Victorian Eye and Ear Hospital are committed to making a real difference for eye health. The strength of the partnership between these three mighty organisations relies on the efforts of many people. At CERA, we've been really fortunate to have had an outstanding and engaged board, led by our chair, Olivia Hilton. Olivia's been on the board for almost 11 years, with almost 6 years as chair. CERA's had the benefit of Olivia's prodigious expertise in commercial and not-for-profit sectors. This includes supporting organisations that drive social change. We've sincerely appreciated Olivia's guidance and expertise. Please join me in welcome Olivia Hilton to the stage to introduce the 14th Annual Gerard Crock Lecture.
Thank you, and thanks, Peter. That was very kind. It's wonderful to be in this theatre here at the Eye and Ear Hospital. I think for many years we never thought this might happen, but well done, Brendan and Simon. You survived the rebuild, and it's fantastic to be here in person tonight with you all. I'd also like to acknowledge the traditional owners of the lands that we're on tonight and pay respects to elders past, present and emerging, and also acknowledge any Aboriginal or Torres Strait Islanders in the audience tonight with us. For those who can't see me, I'm also standing at the lectern wearing a blue blazer, a blue and white striped top, and I have medium brown length hair. I'm also in the naval officer. This is not planned. It is an honour to be here tonight, as mentioned, in person with many of you. And it's also wonderful to be here at the Eye and Ear, and thank you, Shireen, who I know can't make it tonight, and Brendan for the opportunity to use this lecture theatre. One of the highlights for me in the past year has been establishing the joint CERA and Eye and Ear Hospital strategy. That partnership, innovation and exceptional care aims to maximise the benefits of having world-class hospital, medical research institute and a university department under the same roof. We're excited by the many opportunities this creates to deliver better care for patients by integrating research across all clinical specialties and roles throughout the hospital. And we can already see this in action through collaborations on projects like the Bionic Eye, our joint venture on the Lion Eye donation service, and the growth of ocular genetics clinics and pioneering gene therapy surgeries. It is often said that there's never been a more exciting time to be in eye research. At CERA, we believe this to be true right now. We have ambitious plans to work with our research partners in Victoria, Australia, and around the world to make the most of this exciting era. But I don't want to steal Keith's thunder, and we'll let him fill you in on those plans. So let me introduce you to Keith. One of the great strengths of CERA is the calibre of the researchers in our team. From the many outstanding University of Melbourne students who choose to undertake postgraduate research at CERA, to the talented early and mid-career researchers working in our labs and clinics, to our principal investigators and accomplished scientists who are part of the CERA's executive leadership team. Tonight, I'm very excited to congratulate two of our leadership team, Lyndall Lim and Peter van Wijngaarden, on their well-deserved promotions to full professor at the University of Melbourne this week. Well done, Lyndall. It's indicative of the great depth of our team and the skills they bring to work every day in pursuit of CERA's work towards a world free from vision loss and blindness. It is now more than 6 years since the CERA board and the University of Melbourne embarked on a global search for a new Ringland Anderson Professor of Ophthalmology and Managing Director of CERA. Professor Keith Martin, formerly of the University of Cambridge, immigrated to Australia and began his new role in January 2019. Keith came to us with an impeccable research credentials. Like Professor Gerard Crock decades before him, Keith was also a Professor of Ophthalmology appointed to University of Cambridge in 2010. At the time, Keith was also the youngest full professor at the University of Cambridge Clinical School. Keith is also former Deputy Director of Cambridge John Van Geest Centre for Brain Repair and an affiliate principal investigator at the Wellcome Trust, MRC Cambridge Stem Cell Institute. Keith's groundbreaking preclinical research has demonstrated the potential of gene and cell therapy to reduce optic nerve damage in glaucoma. And he is not only a brilliant researcher, but also has a passion to practical application for translating his findings into new treatments for patients. He was the co-founder of the Cambridge-based gene therapy company, which currently has a treatment for glaucoma progressing to clinical trials. And we are extremely fortunate that he has brought that drive for innovation and making a difference to CERA. Early in his tenure, he led the development of a new strategy which has a strong focus on boosting CERA's clinical trials capability and bringing new treatments to patients. The outcome of the work can be seen in the recent deal struck between CERA and Breakthrough Victoria to develop a new world-leading eye clinical trial centre here at the Eye and Ear Hospital, and also work to develop a new genetic engineering hub nearby on Victoria Parade. I will leave Keith to tell you more about those plans later. Keith has been an outstanding leader at CERA and as Ringland Anderson Professor of Ophthalmology over the past 5 years. Both myself and my fellow board directors have worked with many leaders, and I speak for all of us when I say it is an absolute privilege for us to work with someone like Keith. He's one of the best leaders I've seen in action: visionary, strategic, open-minded and curious, collaborative, and above all, authentic. And I'm excited to be able to share tonight that Keith will continue as CERA Managing Director for another 5 years. I'm thrilled that Keith will be at the helm to keep driving our strategy forward to make a difference for people with vision loss and blindness. Thank you to everyone for attending tonight. It is now my great pleasure to introduce Professor Keith Martin, CERA Managing Director and Ringland Anderson Professor of Ophthalmology, to deliver the 14th Annual Gerard Crock Lecture.
Well, thanks, Liv, and that's a spectacularly kind introduction, and I really appreciate it. I'd like to acknowledge the traditional owners as well, and their elders past and present. And for those of you who can't see me, I'm standing at the lectern wearing the regulation blue suit and looking slightly less crumpled than is customary for me. So there's an image of me on the screen. Well, there's not, actually. There would have been. And I'll make sure I try to talk you through what is going on if you're struggling to see the slides as well. So it's an absolute pleasure to be invited to give the 14th Crock Lecture. I was a cheap date, no international airfares required for this one. And when I was wondering about what to talk to you about tonight, I got to thinking about the breadth and depth of the work that we do at CERA. It's a really exciting time at the organisation at the moment, with a major redevelopment currently underway to set us up for a sustainable and successful future. And so rather than just give a lecture based on my work, I wanted to really this to be a celebration of a variety of work currently going on at CERA and how we're trying to achieve real-world impact for people with vision loss and blindness. So I'm going to structure this by looking at some of the transformational technologies we're using to try to beat blindness in a whole range of different ways. And sadly, I can't cover all of the work that we do at CERA today, and so apologies to those that I don't mention. We'd be here all night and into most of the rest of the week if I were to do that, but I hope it gives you a flavour of the sort of work that we do. So let me start by telling you a little bit about CERA, and many of you are friends and supporters already, so you know a lot of this. We're a not-for-profit medical research institute based here in the Eye and Ear Hospital. We're closely and very proudly affiliated with the University of Melbourne, and we rank very highly in the international rankings of vision research and ophthalmology. We're the only WHO collaborating centre for prevention of blindness in Australia, and we're also the largest ophthalmic clinical trials unit in Australia. And we have a very simple mission. We work to eliminate the major causes of eye disease and blindness and reduce their impact on people's lives. And we're based here at the Eye and Ear Hospital, where we are now, and at the Eye and Ear on the Park. But we have big plans. And as Olivia mentioned, we're in the middle of a really exciting redevelopment, which is currently underway, to create state-of-the-art new labs and a new home for our research support staff at 200 Victoria Parade. So we will be the top floor of this fantastic new world-class building, and we will also be redeveloping, and I'll show you the plans as well, a major new clinical trials centre within the hospital where we are. This is a little teaser for those of you who are going to be working in the new facility. This is our Discovery Research Centre, and we're hoping that this will also function as a genetic engineering hub for the local community as well. And we really want to see university partners and other partners with us in this space, because as you'll see today, a lot of the technologies that we're developing are not just capable of being used in the eye, they can be used in many different parts of medicine as well. And again, we think this will really help us to recruit and retain world-class researchers. So when we think about the leading causes of blindness, there are a number of different ways to break this down, but I'm going to do it very simply. I'm going to think about 2 causes of blindness in younger patients, and that's inherited retinal eye disease and diabetic retinopathy. And depending on which country you're in, these are often neck and neck, and they cause a massive amount of loss of quality of life for patients, because they often come on quite young in life, early in life, and they cause problems for patients the whole way through life. So they cause a massive amount of blindness. In Australia, it's inherited retinal diseases that are the leading cause of blindness in the working age population. As we get older, glaucoma and age-related macular degeneration really take over in terms of the reduction in vision. Macular degeneration taking away the central vision of patients, and glaucoma often starting with loss of peripheral vision, but progressing. And both of these conditions can cause irreversible blindness as well. In terms of what we do at CERA, I'm not going to go through a whole bunch of stats. I'll just highlight 2 things. In terms of our activity as a clinical trials unit, last year we had 2,300 people involved as participants in clinical studies at CERA, 60 clinical studies, and 40 of those are interventional clinical trials. And in terms of the other sort of things that we do, again, I'd highlight the work of the Land's Eye Donation Service, who provided tissue for 463 corneal transplants and more than 200 other types of eye surgery last year. That's more than the rest of the Victorian transplant program put together in terms of other organs from one institute. And as has already been mentioned, the joint strategy has been a really huge thing, and thanks to Brendan and his team, we worked hard to put this together because we thought it was important. And the theme is innovation for exceptional care, and we worked together to agree our partnership vision and our partnership values with the hospital, which I think has been a great step forward to make the benefit of that partnership really very real. So in terms of the transformational technologies I'm going to talk to you about today, the first one, and it's one that's close to my heart, is gene therapy. Now, all those of us who've ever had a cold or had a sore throat have experienced gene therapy in one way or another, because viruses have had millions of years evolving to push genes into our cells. And we can hijack that machinery as well to do good rather than harm. And so when there's a gene that is defective, we can engineer a copy of that gene that's repaired in the lab, incorporate that into what we call a plasmid, which is a little piece of genetic material that fits inside a virus, and then that virus can be injected directly into the eye in solution. And that virus can then transfect cells, target cells, change gene expression in those cells, and if we've done it right, can improve the function of the retina or the tissue that we're targeting. And we can use gene therapy in different ways. We can use it to replace a defective gene in that way, but we can also use it to upregulate protective genes or edit defective genes, or actually to reprogram cells from one type of cell into another. Or we can make non-light sensitive cells light sensitive. So it's a hugely powerful technology, and I'll give you some examples of many of these over the course of the talk. And gene therapy is arguably further advanced in the eye than any other organ. It's a very mature technology now, and the eye is an ideal organ because a single injection into the eye can target a large proportion of the cells of interest. That's very difficult to achieve in many of the larger organs in the body. We have excellent tools available like those viruses that I mentioned, and this is already in the clinic. So many specialties are talking about using gene therapy. We're already doing it with good safety profile and able to target many of the different types of cell that get affected by different types of eye disease. So in terms of inherited retinal diseases, as I said, this is a major cause of blindness in the working age population and huge socioeconomic impact. And we've made great strides over the last few years in getting the Eye and Ear Hospital and CERA working in partnership to deliver gene therapies as routine clinical practice now. So we're regularly delivering gene therapies, and I'll show you examples of that in a second. We've treated 7 eyes so far, for example, with Luxturna, which is a treatment for a form of retinitis pigmentosa. We have multiple other patients who've been treated in gene therapy trials for macular degeneration, and many following studies for retinitis pigmentosa, recruiting and treatments getting underway, as well as a pipeline of new therapies that we're developing in-house that I will mention to you. And the trailblazer for this has been a drug called Luxturna, which was approved in the US in 2017 and in Australia in 2020. And Melbourne is one of the only 2 sites in Australia where this is delivered. And Tom Edwards, who's in the audience, this is Tom doing a treatment with Luxturna. So what you're seeing here is an orange cannula, which is finer than a human hair, being used to inject the virus onto the retina. On the right-hand side, you see the little bleb of fluid that's raised up as well. So a small amount of this injected. And it's a bit like injecting underneath wet tissue paper, if you like. This is a hugely delicate tissue, and so enormous skill required to do this. But what does that mean for patients? What does it mean in the real world? And this is absolutely a life-changing treatment. I think that's important to emphasize. For most of my career, and many of the rest of the ophthalmologists in the room have been telling patients with inherited retinal disease, there's nothing we can do. And we were all pretty sick of that. It's not a nice feeling. We like fixing stuff as ophthalmologists, and we couldn't fix this. But let me introduce you to a little 4-year-old boy who was the most recent person treated with Luxturna in this hospital just a few weeks ago.
Step up. Good job. Good job. Stepping down.
I think not too scared.
Yeah, don't be scared. Come on, you can do it.
Too scared of dark.
I know.
Yeah, we're gonna come.
Too scared of dark.
Here's cat's
So we have to light.
Okay.
Light.
Okay.
Mama will turn the light on once we finish. Here's orange. Yeah, you're okay.
So you can see he's feeling for the, he can't see, so he's feeling with his feet.
Here is red.
Not even trying to look down at the steps.
Here is blue.
And this kid had to feel his way around the house, around the furniture in low light conditions.
That's it. Bumpy, bumpy, bumpy. Keep going.
And again, not looking down, just using tactile perception from his feet to navigate.
You are so...
This is 3 weeks after his gene therapy treatment here.
Well done. Stop it. Now come back again.
I'm the winner.
You're the winner.
I'm on the way.
Oh, be careful.
I am on the way.
So life-changing treatment. So how do we match patients to these treatments? Well, that's a really important part of the process because we need to find these patients. And as part, that's been a really fantastic collaboration between the Eye and Ear Hospital and University of Melbourne researchers. And you see Laura Nathan and Tom Edwards, who are in the audience here today. They lead our gene therapy unit and have driven the creation of an ocular genetics clinic that streamlines patient pathways for patients with inherited retinal diseases. And so rather than having 2 separate services, a clinical service and a research service, they've brought this all together so that patients coming in are assessed, they're consented for being recruited into clinical studies. If there are treatments available, they're streamed to those treatments. If not, they become part of natural history studies. They're genotyped and phenotyped and part of a database of patients who are ready to go when these new treatments come along. So a really good example of the hospital and the institute working together to improve patient pathways. And as well as developing new treatments at CERA, a key part of what we do is to provide the evidence that they work in the real world. And as has already been mentioned, we're the largest clinical trials centre in Australia. We can run all phases of clinical studies, but the problem that we had was that we'd outgrown the capacity within the hospital, and that was limiting our growth. And so we're delighted that after a huge amount of work by our team, we were able to negotiate with Breakthrough Victoria, and they made a large investment of $10 million in helping us to create a state-of-the-art clinical trials centre on this campus. The first investment, I think, that they've made with a medical research institute. And thank you to the Breakthrough Victoria team, some of whom are here today. We believe this was a huge vote of confidence from the Victorian government in what we're doing and our capabilities. And also a big shout out to Robin Geimer and Lindell Lim, who have been instrumental in building our clinical trials capacity over the years. and also to Tina Cheng and Rowan Nielsen and Nick Northcott, who worked so hard in the negotiation and business planning to get this across the line. So we believe this will attract many more international clinical trials to Victoria, but also enable us to trial new therapies that are developed in Australia here in Australia, and bring new therapies to patients early, as well as contributing to the economy, and again with the ability through the systems that we're developing to expand telehealth and recruitment from rural and remote areas as well. And this is an architect's impression of the space that we were developing, and we're hoping the artist's impression of the space is more accurate than their rendering of a guide dog, which is somewhat less convincing, but we really have fantastic facilities here to do the sort of clinical trials that we want to do. So I guess I should talk a little bit about my own personal research and how that fits into what we're doing at CERA. And my interest is in glaucoma. And to help explain what that is, I'm going to use a 3D model developed as an educational tool for patients at CERA by Associate Professor Elaine Chong, supported by the CERA Innovation Fund. So this is a 3D rendered model that we can use now to educate our patients. So we often think about glaucoma as a high pressure in the eye when fluid produced in the front of the eye can't drain as freely as it should. But actually the main issue is damage to the optic nerve at the back of the eye, and that's what causes the gradual loss of vision, often the periphery, that progresses centrally, that causes the disability that our glaucoma patients experience. And for many of those patients, lowering the pressure in the eye is sufficient to prevent them from going blind. But the focus of my work for a number of years has been on what we do when reducing the pressure is insufficient to prevent blindness. And about 15% of patients who are effectively treated with pressure-lowering treatments will go blind in at least one eye during the course of their disease. And so that's the focus of my work. And to cut a long story short, the work of my group over the last 20 years has led to the development of a candidate gene therapy that is extremely effective at preventing optic nerve damage and visual loss in animal models of disease. And all these little red blobs are the sort of work that we do to count the effect of this treatment within the eye. And we could rescue about 70% of the cells in the optic nerve that would otherwise have died by using this gene therapy treatment. And that has moved on to industry now to take this forward. So there's no way you can bring a new gene therapy through to clinical use with purely grant funding alone. And so we were fortunate to be able to partner with a major pharmaceutical company who are taking this treatment forward to the clinical trial stage. The other way that I'm using gene therapy in my work is to not just to slow down the rate of decline, but also to try and restore vision in glaucoma once it's been lost. That's the holy grail of glaucoma treatment. And as part of that, a lot of my work in Cambridge was looking at why the spinal cord and the optic nerve and parts of the brain are so bad at regenerating. When we injure a sensory nerve in our skin, it regrows. It finds its target, it regrows back again, and yet the optic nerve can't do that, and the spinal cord can't do that. And so the basic science is here to try and find the mechanisms why that occurs. What's the difference between a sensory nerve and an optic nerve or a spinal cord nerve that explains that difference? What's the difference between them? What's the difference between when our eye is developing and when we're an adult? That means that the nerve fibers can grow when we're developing, but not when we're an adult. What are those differences, and can we harness what we know about those differences? And so again, just to give one example of that, we found that one of the major differences between developing optic nerve and adult optic nerve was the absence of this molecule called protrudin. Now, in science, the clue is often in the name. And protrudin helps structures protrude, be they cells that are trying to migrate or nerves that are trying to regrow. And it brings all the machinery that you need to regenerate to the right place at the right time. But it doesn't happen in the mature optic nerve. And so normally when you cut an optic nerve cell or a retinal cell in the dish, it doesn't regrow. And so we could do this in the dish. And we could use a laser to cut those fibers, and normally they wouldn't regrow unless you treat them with this gene therapy, in which case they remember how to regenerate, and so they can grow for long distances. And from starting to do this in the dish, we were then able to do it in animals with injured optic nerves as well. So we can now regrow the optic nerve after crush injury all the way back to the brain to establish connections back in the brain. simply by teaching these nerves something they've forgotten over the course of development. In terms of other ways that's a long way from the clinic. Other projects that we're doing are much closer, and so there are other ways that we believe we can slow down glaucoma tomorrow if we can demonstrate that they actually work. And Jonathan Croston, my predecessor in this role, who many of you will know, and Flora Hui, who's again in the audience tonight, led some really pioneering work, first in human studies, to show that using vitamin B3, it was possible to improve some aspects of function in glaucoma patients in the short term, simply by taking this vitamin, which has a profound effect on mitochondria, which are the energy-producing components of the cell. And off the back of that work, we have led an international collaboration, which has been formed and is coordinating a very large effort in multiple countries across Australia, Sweden, and the UK. We all now have funded trials in those different countries, over 1,000 subjects to be recruited to answer this question once and for all, if this treatment can improve the outlook for glaucoma patients with a very simple, very cost-effective type of treatment. And I'm sure there are people in the room who are actually participants in that study. Say it will be over 1,000 patients, and it will be the second largest glaucoma neuroprotection trial that's ever been attempted to do. And so we're recruiting actively. We have more than 90 patients recruited to this in Melbourne already, with the study in Sweden underway, and the study in London, in Murfields, getting underway in the early part of next year. So if we change tack a little bit, another question is whether the technologies that we use can improve routine day-to-day eye care. And I'll give you a little example of that, again, giving credit to Jonathan Croston and also to Hugh Taylor's daughter, who is also in the audience tonight. And Kate was instrumental in helping this project get underway, because they spotted a problem that the communication between optometrists and ophthalmologists in Australia was pretty terrible. Scans were being faxed back and forwards that were illegible. There was very little information trail, and they said, surely this can be improved with technology. And so within 5 years, they developed a telehealth system which was being used by 70% of all eye care providers in Australia and New Zealand. It was expanding into the US and the EU. And during the early stages of COVID, it was acquired by a large international company, allowing us to recycle some of the profit of that into supporting new innovation at CERA. So a real telehealth success story, but it's also a research tool for us, and it also helps to improve patient care in other ways. And a real example of that is that it's helped solve a problem in diabetic eye disease, in that screening in Australia through a program called KeepSight, pioneered by Peter van Wijngaarden, who introduced the meeting this evening, is now being used and this is being used to improve diabetic screening. And this is because 50% of the 1.7 million Australians living with diabetes don't have access to regular screening. A third of signs of diabetic eye disease, and most of that vision loss is avoidable if we catch it early enough. So again, using one right eye, what we're trying to do is pick up diabetic retinopathy at the early stages, when there are subtle changes in the capillaries of the back of the eye, and prevent the deterioration that will inevitably occur if this is untreated to proliferative diabetic retinopathy, with hemorrhage and bleeding and scarring, and profound loss of vision. And KeepSight is really part one of a solution. So 1.2 million Australians with diabetes are registered with the National Diabetes Services Scheme, and KeepSight is digitally integrated into their health patient management system using Oculo that automatically sends reminders when next health checks or eye checks are due. And so this is really taking off. More than 600,000 eye checks so far on the system, building in regional and remote areas with dedicated programs to engage Indigenous Australians. So a real example of the sort of technology developed in-house having a real-world impact in Australia and further afield. But we were talking about gene therapy. So can gene therapy be used to treat diabetic retinopathy? And diseases where new blood vessels grow in the eye are a real problem. Diabetic retinopathy is one. Macular degeneration is another one. And we've been hugely successful in developing new treatments for some of these over the last few years that have revolutionized these treatments. But those treatments require many injections into the eye, and I'm sure many of you will know people who are having treatment for macular degeneration that have monthly injections into the eye for years on end, dozens and dozens of injections. Think about that in terms of the inconvenience and discomfort and infection risk for patients and also for health systems. This is a huge burden for health systems. And so Rick Liu, again working at CERA, has developed a specially engineered virus to develop the therapeutic gene anti-VEGF into retinal cells as a once-and-done injection. But the beauty of what he's developed is that he can then switch that. So the technology is called switch gene. And so the therapeutic gene can be switched on and off with a simple eye drop. So this removes the need for repeated eye injections and reduces the burden to the patients and the health system. So a potentially transformative treatment which is under development right here. In terms of other ways that we can use genetic engineering, another one that I'll highlight is cell reprogramming. And many of the conditions that cause blindness are due to photoreceptor loss, the loss of the light-sensitive cells within the eye. And that includes retinitis pigmentosa and macular degeneration and many other conditions. But the problem is there's no cure for blindness once photoreceptors are lost. The child that was treated, that I showed you the video of, we treat those children early before they've lost photoreceptors. But if the photoreceptors have gone, that type of gene therapy just doesn't work. And also that type of gene therapy, because there are more than 300 different genes that cause inherited retinal diseases, we believe we're not going to get 300 different gene therapies. And so we need approaches to genetic eye disease where one treatment can be used for many different eye diseases. And so that's what we're trying to develop. And so Ray Wong, working here, has been working on a project to reprogramming non-photoreceptor cells in the retina into new photoreceptors. Now this may sound like a crazy thing to do, but it's exactly what happens in fish and amphibians after injury to the retina. So they have the ability to regenerate these cells, and we've lost it somewhere along the lines of evolution. And so Ray's work has been predicting computationally which transcription factors would be able to restore that regenerative ability within the eye. And this is a technology that has been patented by CERA and is working 100 times more effectively than the leading competitors in human compared to what others have been able to achieve in mice to date. And again, as a story of how we do things, that was a project supported by our in-house innovation fund that then went on to get nationally competitive grant funding through the NHMRC and other bodies, and then has recently been picked up by the Medical Research Futures Fund, curator funding, leading to the spin-out company Myriagen, which is currently exploring partnering opportunities. So in terms of the progress we're making, this gene therapy is really pretty effective for a few diseases so far, but we're broadening it out and using it now for glaucoma, macular degeneration, and diabetic retinopathy. And we're also developing cell reprogramming techniques that harness gene therapy to reprogram cells to restore vision. I'll give you another example of a technology that's being developed in-house, and this is again the work of Peter van Wijngaarden. And this is exploring new ways to image the eye. Conventionally, we image the eye using white light, and white light gives us a color photograph of the eye that doctors can interpret to look for signs of disease. But if you remember your school science, white light is made up of a whole spectrum of different colors. And the way in which those colors are reflected or absorbed by tissues of the eye contains a lot of information. So there are different processes that occur in the eye that change the absorption characteristics, and we can interrogate that using a scan of the eye. So it's a bit like the difference between, for example, the Hubble telescope, which uses visible and UV light, and you can see what you think is amazing detail of the universe, until you look at what the James Webb telescope can do using infrared as well, and look at the amount of extra information that's in there. And that's sort of equivalent to what we're doing. We're making use of all of that extra spectral information to show us enormously more about the eye, things we've never been able to see before. And that's not just eye disease. So one of the really exciting aspects of this technology is the amyloid that accumulates in the brain in Alzheimer's disease also accumulates in the eye. But we can pick it up in the eye in a scan in a fraction of a second rather than the invasive techniques. And if we can pick it up before the brain is too far gone with Alzheimer's disease, then the new generations of disease-modifying therapies that are coming through could potentially be of benefit. So I think that's a really exciting development, and the camera is outside here if you want to have a look at the actual camera, which is now moving through the testing phase. So again, another really exciting use of technology. The final one I'm going to mention is for age-related macular degeneration. And again, we've had great progress with the anti-VEGF treatments for wet macular degeneration. We haven't had the same sort of advances for dry macular degeneration as yet. And like most neurodegenerative diseases, most treatments are more effective if you can get them in before you've got a lot of loss of function and a lot of damage to the structures. Once the brain is scrambled, once the eye is scrambled, it's really, really hard to repair. So you really want to prevent it from getting that stage if you can. And again, using one right eye, again, thank you, Elaine. What we're trying to do here with the macular degeneration treatments is prevent the early stages of macular degeneration getting to this late atrophic stage of macular degeneration where there's a large area of loss of function and central blindness, or where there is this more leaky and hemorrhagic scarring response which occurs at the back of the eye. And so there's a real unmet need for treatments that slow down the progression of early to late macular degeneration. And Robin Geimer has been pioneering the use of what we call nanosecond subthreshold laser, so low energy laser treatment to prevent this deterioration. And what Robin has shown is that in a subset of patients with macular degeneration, that you can classify using a very straightforward scan that we use in all eye clinics. If you pick the right patients and treat them with this laser, you can prevent this progression from intermediate age-related macular degeneration to the advanced stages of macular degeneration. And Robin is currently organising a large clinical trial with this Australian-developed technology to test this in the real world. So really just to summarise, when we're thinking about transformational technologies to beat blindness in the younger age group, in the inherited retinal diseases, I've shown you some examples of gene therapies and cell reprogramming. I didn't even have time to talk about the bionic eye, which has been the subject of a previous Crock lecture from Penny Allen, who is its pioneer. In terms of diabetic retinopathy, again, I think the switchable gene technologies are really exciting. We can use hyperspectral imaging to pick up features that reduce the need for invasive angiography, for example, and we can also use telehealth to improve the care of these patients. Again, in glaucoma, we have gene therapy and cell reprogramming and vitamin B3 and hyperspectral imaging, and in macular degeneration, these approaches, but also this nanosecond laser technology, which I think is really promising. So overall, I think in terms of what we're doing at CERA, I'm really proud of the work that the teams are doing here, and I'm sorry I couldn't show you more of it in half an hour today. I think we've built a really strong culture of innovation and translation. I'm really proud of the joint strategy that we've developed with the Eye and Ear Hospital and our strong partnership with the University of Melbourne. Many of us are joint appointees between the university and the hospital and the institute, and I think that adds to the strength of what we do. We're the most active clinical trials centre, but we're also building the capacity to double that sort of activity with the new clinical trials centre that we have, and we have a strong pipeline of other projects aiming for real-world impact. And we see ourselves as really being part of a growing, exciting biotech infrastructure within Melbourne. There's a lot of work going on in different disciplines. We want to be at the heart of that, and we want to share technologies, learn from other technologies, and build innovation hubs around these sort of technologies so that they can be shared with other branches of medicine as well. And of course, we have an amazing community of supporters as well that help to make this happen, as well as the researchers. And I think we try to be an example of how a medical research institute and a university and a hospital can work together to advance eye care. So it's been a real privilege and honour to give this lecture today in memory of Professor Gerard Crock, and I'd like to thank you for all of your support and for your attention this evening. Thank you. So I thought we'd do something a little bit different with the Q&A today, and as a lot of the work I've been talking about tonight is the work of others that I'm shamelessly showing off, I'd like to invite some of them onto the stage to answer some of your questions tonight as well. So firstly, I'd like to introduce Professor Robin Geimer, AM. So Robin is CERA's Deputy Director, a Professor of Ophthalmology at the University of Melbourne and a retinal specialist here at the Eye and Ear Hospital. And Robin is, of course, a world-leading macular researcher and was just last week named the nation's top-ranked ophthalmology or optometry researcher by the Australian Research Magazine for 2024. So superb. And next, I'd like to welcome back Professor Peter van Wijngaarden. So Peter heads up our ophthalmic neuroscience research at CERA and is head of a spin-out company Enlighten Imaging, which is developing the eye scanning technology that I showed you. So coming up next to sit with Peter at the front is Professor Linda Lim. And so Lindell is head of CERA's clinical trials research and leading researcher in diabetic eye disease and uveitis, and a professor in the Department of Surgery at the university, and also head of the ocular immunology clinic here at the hospital. She's a busy lady. And in the moment, I'm going to move to the chair next to her at the podium, and replacing me here will be Associate Professor Lauren Ayton. So Lauren is head of the Vision Optimization Unit in the Department of Optometry and Vision Sciences at the University of Melbourne, and she's also a joint principal investigator of the Venture study that I showed you, a collaboration between the University of Melbourne and CERA to understand the progression of inherited retinal disease. And as well as being a great scientist, Lauren is also an exceptional communicator. She's the regular host of RRR radio program Einstein A Go-Go, and we'll use some of those skills to moderate our discussions tonight. Lauren.
Great. Thank you, Keith, and thank you again, Keith, for such a fantastic talk. It's really quite inspiring to see what is achieved at CERA, and I think we're all very proud of what we're doing here. So this is your opportunity to ask some questions. So we have Rich and Callum in the audience with the microphones. So if you put your hand up, if you have a query, they'll come to you and you can ask it out loud. But while you're thinking of a question, I've got a few of myself. And one of the statistics that really stuck out to me in that presentation was that we've seen over 2,000 clinical trial participants in the last year, which is an outstanding number. And Lindell, you head our clinical trials unit. And you must be incredibly proud of that number.
Yeah, go team.
Very busy. So with that number of participants, how do you find that many people, and what should people know about being involved in clinical research?
Yeah, so I'll tackle that first part first. We actually attract referrals from a whole lot of different sources. So first up, we've been doing trials for a long time. We have a great reputation, and so we do have external referrers, some in the audience who actually do refer their patients to us for eligible trials. The second, as a clinician, one thing that I've always really valued is the fact that I can tell my patients not only about the treatments that are available now, I can also actually say, well, actually, there's also a trial of a new treatment that might be better. We don't know, but are you going to be interested? And thirdly, all of us here, what we also do is we often, obviously, speak at conferences, and this is the time that we can actually spread the word to our colleagues who are also practicing in other areas to say, look, this condition that you thought that wasn't curable or didn't have a treatment, we now have a trial of a new treatment that actually might be useful for your patients. Please spread the word, tell your patients, and refer them in if they might be eligible. So that's the first part of the question. What was the second?
I guess the second is for anyone in the audience that is thinking about being a participant in one of these trials, what does it actually entail? What do they need to know before they come in? Yeah. What will they expect?
So certainly I would say that I tell all my patients they are special, but I think trial patients are extra special. Mm-hmm. And I'd like to say that my team certainly does look after them really well. And what you expect is basically you have a full assessment, we talk you through what the trial might entail or the study might entail. It sometimes includes a treatment, sometimes it doesn't. And we do ask that you come in on a regular basis. So there's usually some follow-up visits. Now, some of those might be every month, and a lot of people might sort of sit there going, every month, for how long? But if you actually think about it, for instance, for things like age-related macular degeneration, some of you might actually be needing to have an injection every month anyway. But in your trial, you actually can get a whole year's worth of appointments in advance, which is actually great to actually plan in ahead, particularly if you've got a job or something. You can actually say, well, this particular day for the next 12 months, I'm not going to be here. And that's actually really helpful for forward planning. But we, of course, can also, there's a bit of wiggle room as well, always. But also, you also see the same team every time, same team members. It's, I would say that that personal approach has actually helped us keep our patients throughout the COVID period as well. And I think that speaks a lot to the confidence that, or the relationship that we have with our trial participants.
Yeah, absolutely agree. And I think that's a big strength of CERA, is that the research participants become part of the research team. You know, they're interacting with what we do. And I wanted to hand over to you, Robin, now, because the other really important people in our research is industry and companies that we work with. And you've got an amazing track record of working really well with industry to design research. Keith mentioned the laser study and what we have done so far in Australia. Where to next with that?
Thanks, Lauren. So I think it used to be sort of a no-no to talk to industry, and it was very sort of something that you weren't supposed to do. But I think we've moved past that and realised that together we can do much better. And in fact, a couple of years ago, I spent a whole year at Roche in Basel, realising that there were things that I wanted to do that I couldn't do by myself. And so I think the laser is a good example of an Australian-made technology. We're very good at making lasers in Australia. And so together we've been able to approach venture capital, we've been able to go to the FDA, find out what they need to try and plan the best trial together. And I think industry has deep pockets to be able to give them my expertise in how to actually plan an earlier trial. And so together, go to the FDA with plans that one or other of us wouldn't have come up with ourselves has been a very useful way to go forward.
Absolutely. And I think your knowledge, and especially from you spending that time, is very much spreading across CERA as well, which is benefiting us all. So you can probably tell I could talk all night. So does anyone else have any other questions for our esteemed panel? Wonderful.
I'm not sure whether to direct my question to Keith or to Peter, but perhaps via you, Keith. Obviously, hyperspectral imaging is a really very exciting technology, and I've sort of watched it evolve over the last few years here at CERA with some great anticipation. And I, by the way, I love your analogy about it being likened to the James Webb Telescope and the ability to generate greater detail about things. But I wondered what you see as the role of hyperspectral imaging in glaucoma, because traditionally we have been using, for those that don't know, optical coherence tomography to measure the thickness of nerve fibre layer and ganglion cell layer and other things. What do you see as the role of hyperspectral imaging? Do you think it will replace OCT, for example?
No, I don't, and it's a great question. I'm sure Peter could address it just as well. But we're looking for new endpoints in glaucoma to shorten glaucoma clinical trials, and that's a work that is being funded by Roche as part of another partnership independent of anything that we're doing with macular degeneration, using hyperspectral imaging. And what we're doing is, when you have a field defect, so you have an area of damage to a sector of the optic nerve, the most likely place that further damage will occur is on the edge of that area. So if you have a shadow, it's in the penumbra around that. And really excitingly, what we're seeing with hyperspectral imaging is, we think, is the ability to pick up, sick tissue around the dead tissue. And that's really important as an endpoint, because if you can reverse something which is sick and make it better again, that's a very different endpoint to slowing decline over many, 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 many years. And it's the difference between taking something and just trying to measure a rate over many years by saying, well, something that never happens, which is this sick tissue recovering, is happening. and therefore we've really got something here with this new treatment. So that's one example of how we're looking to use hyperspectral imaging in glaucoma, and there may well be others, because almost everything that you're looking at, you're finding new things that we just couldn't see before, and then it becomes how we interpret them and which ones are useful and which ones are going to change the way we do things.
And I might just add a little plug here for Zichao Wu, who's one of the principal investigators who's leading that glaucoma study and really making waves in many other fields of imaging research in glaucoma and AMD.
Absolutely.
There's a question here.
My name is Jean Miller, and we'd be very excited to be involved with Keith Martin for some time. But I was just wondering if I could ask a question about young children with glaucoma, because there does seem, I've heard of a few children that have been involved with that. Is there any particular advance in trying to treat young children?
That's a great question, Jean, and I've over many years have treated children with glaucoma. It's a difficult area to work in. It is one of the areas, though, where early surgery can be transformative. And surgical techniques have improved over the years. And because in young children, it's very much a pressure-dependent disease. So in some elderly people, the nerve is vulnerable and the nerve is weak, and that's another problem. Most of the childhood glaucomas, it's the pressure is the problem. If we can solve that problem, they have the chance, if we get in early enough before there's been too much damage, of them having good vision for the rest of their lives. And we see that with the primary congenital glaucoma, the one that comes on very soon after birth. If we treat those kids effectively with surgery, 90% of them will do very well. And so it's really getting in early enough to do it, and the advances in surgery have meant that we can do those treatments more reliably with the type of surgery. So the hope is that they don't need necessarily these gene therapies as long as we get them early enough. If we don't, then we're then back into needing the regeneration and the other things that we were talking about today. But there have been great advances in the management of pediatric glaucoma over the years.
Sons with a malignancy of the histocytes, and they said one of the things that would have happened, he would have lost his sight. Is that correct, or?
Well, I guess I don't know all the details. That's a terrible story. But there are different ways in which different types of tumour can affect the eye, and there's quite a lot of crossover in that situation, but yeah, that's terrible to hear.
Great. Any other questions before I jump back in? Up in the middle, fantastic. And I think just following on from what Keith said too, I think this is one of the really other exciting things with CERA. The eye is basically an extension of the brain, so we can see things in the eye which we can then link back to things like Alzheimer's, so it's incredibly exciting for our research.
Thanks very much. And I found it really inspiring to see a lot of the really sort of grand new transformational technologies that are solving problems that we always thought were pretty intractable. So I guess my question is, what's next? What are the new big problems on the horizon that are preventing these technologies from reaching their full potential that we haven't solved yet?
Funding. These are very expensive technologies to get to the world. So I said that in jest, but there's actually some truth to that. We're great at generating rigorous evidence, but we're still learning about how to engage with industry, how to leverage venture capital. There is an evolving ecosystem, which the University of Melbourne is playing a key role in to really stimulate that environment. But I don't want to hog the microphone.
No, absolutely. And I think, a lot of these things that start out being extremely expensive, as technology moves on, become everyday. And, the Human Genome Project, for example, sequencing the first genome cost billions of dollars. Now you can send off your 23andMe, and for a couple of $100, you can get much of the same information, on yourself
for a couple of $100s. The gene therapies that we're giving at the moment, that gene therapy, that Luxturna treatment at the moment, close your ears, Brendan, costs $800,000 per treatment in the US. And the reason for that is because this is a really rare condition. There are probably only 20 people in Australia that have this condition, and yet you're trying to recoup the cost of development, which has taken 20 years to get to that stage. If you're dealing with a condition like glaucoma or macular degeneration, which is hugely more common, the economics are completely different. So the cost to produce these vectors is coming down all the time. I would expect the cost of goods for these treatments to get to a few $100s within the next few years, not hundreds of thousands of dollars or millions of dollars, a few $100s in terms of the cost of goods. So that's the direction of travel. Technology and economies of scale and other things change what is 1 in a million sort of ability to do something to something which becomes routine quite quickly, and that's we're pretty creative primates, and we tend to find a way to get these things done. So I'm optimistic that technology, if we apply it appropriately, will solve many of these issues.
One of the issues we have before we get there is how do we actually deliver what's currently in the pipeline coming. So for example, with dry age-related macular degeneration, we're about to have the first 2 treatments that we don't have a treatment for, and then a third and a fourth and a fifth, but we have no ability to treat. There's no room to treat this aging population. So as the population gets older and we're not keeping up with ophthalmology trainees to deliver the care. So whilst it is currently quite labour intensive, until we get gene therapy, we've got this little time zone where the diabetic explosion, the ageing population, we have treatments, but to get them in the door to give it is currently a very practical problem.
Yeah, and it's a really important point, Robin. And our experiences from KeepSight, we shouldn't need KeepSight in Australia. We've got a great health system. We've got plenty of eye care providers. We've got free eye exams under Medicare. We've got the best access to PBS pharmaceuticals, and yet 50% of people are missing out on eye checks. And that's because healthcare is more than just technology. It's about a system, it's about integration, and it's about humans. And so, you forget about the system at your own peril. So that's been a very important learning for us.
Absolutely. So with an eye on time, we have time for one lucky last question. Over here, thank you.
Charles Masek, a founding director of CERA, and had the pleasure of working with you in the early days as chairman for a few years. So it's wonderful to see what CERA has become. My question is a generic one and can be to any of the panel. What potential does AI have for accelerating some of the development and evolution of medical research, particularly in this space?
Peter.
Yeah, look, I mean, a lot of our diagnostics utilise AI. I mean, I think it's a technology like others. There's a lot of hype around the technology. There's a lot that we don't yet understand about how these smart decisions are being made. So I think they have huge potential. They have huge potential in clinical care more broadly, annotating notes so that clinicians could actually spend more time having meaningful interactions with their patients, helping to standardise care delivery, huge potentials in managing the large volumes of data that we generate. Every clinical episode now is becoming a big data challenge. Even our standard imaging technologies generate vastly more data than any human can interpret. This is, in the average eye clinic. So it can help distill out the noise and allow the clinician to focus on what's important. The sky is sort of the limit, and I think we just need to be careful that we walk before we can run in the implementation.
It's a beautiful spot to finish up our panel discussion today. So we obviously didn't have time to get to all of your questions. So if you have anything else that you think of later, you can always reach out to us at community.cera.org.au, or obviously reach out to any of the researchers at CERA. We always like to discuss our work. Please join me in thanking the illustrious panel for their discussions. And I will just pass on to Peter to finish up the evening.
Thanks, Lauren. I'll be brief, I promise. So look, I'd like to congratulate Keith on such a memorable and inspiring lecture. So much achieved and obviously so much ahead, and I think that's just tremendously exciting. To members of the audience, I think you'll agree that it's just so exciting to be part of this community and to see the hope that lies ahead of us. We're all brought together by our common sense of purpose to reduce vision loss and blindness. To conclude the evening, I'd like to invite Marnie and Trish, two of Gerard Crock's daughters, to present Keith with a small token of our appreciation.
Thank you so much.
Thank you very much. So everyone, enjoy a safe trip home. Thanks for coming.